Give me a second. So we are going to talk about today data search and pairwise alignment. So let's start from an interesting point where this coronavirus. So these days coronavirus, we talk about that a lot, and still these bioinformatics work a lot on finding characteristics and some similar proteins in this coronavirus genome. Okay, so this is one of the attempts. So this is actually downloaded. Actually, this is taken from a very popular journal. This Frontier. I think you can't see my screen. Can you know? So I'll share the screen. Share the screen so that you can see. Okay. So I think now you can see the paper. So it's Frontiers in Immunology, which has a very high impact factor. So here in this paper, I found this particular figure which shows you the phylogenetic relationships. So it means actually what they have done is they have compared strains of this coronavirus. So you know that some people say that this coronavirus is different from that coronavirus which appeared 1 year ago. It's mainly because these different strains appear. The strains mean the genomic sequence is being changed at several places and a new kind of virus appears. Okay, so that happens. I think you may have heard about that disease transmitted by mosquitoes. Okay, so that viruses also change. So previously there was a very, discussion. There was a very huge discussion about this malathions. These mosquitoes are actually adapted to that. Okay, so and the virus would come with the adapted mosquitoes in a different form. Okay, so likewise, this coronavirus also would change the strain. Normally in environment it changes because of the environmental factors, so this genomic sequence can change, and the changes which can survive well with the environmental conditions, that would grow fast. Okay, so that's what actually happens. So we see that the previous strain is not now does not appear, and the new strain appears. Okay, so those kind of things are related with that intermediate genomic sequence. Okay, so so those are things you need to learn. So don't get upset that about that you do not get what I say. So these things. So eventually you will, get familiarized. Okay. So what is phylogeny? So the phylogeny is actually looking at the relationships and looking at the past, how they evolved, some species, how they evolved during a certain time period. Okay, so here also in this figure, you can see the strains of coronavirus, different coronavirus types, in other words, okay? They have been compared. So this comparison is shown as a phylogenetic tree here. Okay, so that's what they have shown here. And if you're interested, you can read that article. So that's very attractive and important at this time. So, so I will first start with that, and the importance of this sequence alignment appears mainly because the other reason is actually it's important mainly because there are more than 400 complete genomes have been sequenced. So now this number has been increased. So I have prepared the slide quite maybe 2 to 3 years ago. So you can see the number of complete genomes which have been sequenced now. So it's increasing day by day. And if you look at the genome database, which we discussed last week, you can see how many genomes are available, okay, which would give you this, all the gene of a particular organism. So these organisms, starfish and these mosquito, dog, rat and mouse and chimpanzee and human, they all are, have been sequenced. So what? So these sequences are, what can we do with these sequences? Actually, we can see similarities in this sequence level. What similarities are available there? So they have found that 99% similarity appears in the genes of chimpanzee and human. Okay, so that's one fact they have found from these comparisons or alignments. So that's what we are going to talk today: pairwise alignments. So these alignments can be pairwise or multiple. Pairwise means we are trying to compare two sequences at once. Okay, and in multiple sequence alignments, what we are doing is actually we are trying to align a set of sequences. Okay, so in this bioinformatics, what we are actually doing for computer science students is actually we are trying to develop computational algorithms and methods used in other computational techniques to actually to present a solution for a particular problem. Okay. So we know what an alignment is now. Alignment is actually put into pairwise alignment is actually put into sequences into two rows and finding similarities in simplest words. Okay. So if it's a multiple sequence alignment, what we are doing is we put multiple sequences one after the other and try to find a similar region, okay, in the particular set of sequences. Okay. So that's called alignment. So we try to align the similar regions of one sequence to the other, okay, and trying to find out similar regions. So so this is very important to find out phylogeny. So in phylogeny also, assume that we have five sequences coming from 5 species: dog, cat, frog, and mouse. Okay, and we are given 5 proteins. Okay, and we know every animal has fur, like you know, different colors for we also have, which is not visible. So if you are asked to develop a phylogenetic tree, what we do is actually we try to find similar regions in those 5 sequences given, and then finding the similarities in between species, and as a whole, if you find a similar region, you can get them into a single root. Okay, you say that there's similarity you can see in the all 5 species. So we can start from a single root, and then we can find out the similarities in between those 2 species, 3 species likewise. Okay, so then we get two species and then join them into one root, and then join that root into the parent root. Okay, so likewise we draw this. You can draw a tree like structure looking at the similarities. So the basis for that development is this alignment. Okay, so up to now, what I have talked with you is the importance of alignment. So it has many applications. Like if you have found a new protein, so assume that you are a biologist and you found a new protein. You don't know where this protein, what the function of the protein. Okay, can you suggest me a way of finding the function of this particular protein? So you know there are protein databases, isn't it? In those protein databases, most of the proteins functions are identified. Okay, so if you can find a similar protein to your own original protein, what can you do? You can predict that this protein also should have the same functionality as my own new original protein. Okay, that's one advantage of these pair alignments. Okay, So in this picture, what you see now, so here you can see these evolution relationships have been drawn. Okay, so this mouse and rat are drawn using a single root. So they are actually two different kind of mouse. So they are related to each other. Okay, so they are in a single root. So this can be found out from the sequence similarities. There's a very high similarity in the sequences of this species and this one. So that's why they are connected into a single root. And here also, chimpanzee and human, those two sequences are also have very large similarities. That's why we have connected that with the single root like this. Okay, so that's the evolutionary application of alignments, and also in epidemic disease viruses. So when they come like coronavirus situation here, these sequence alignments play a very high importance. Okay, they have a very high importance. So in talking about this evolution, so some of the terms you need to learn first. Okay, so first one is homology. What are homologs? Okay, so there are two different types of homologs. So first one is orthologs, second one is paralogs. So what are orthologs? Okay, so if you look at this picture, okay, to understand the orthologs and paralogs, so you can see this at this point speciation occurred. So what is that? New species appeared. Okay, and here at this point, this speciation, you can see again a new uh, species appeared. However, if you look at here, gene duplication event. So here you can see at this corner you have one gene, beta globin, and on this side you have one gene which is alpha globin. Okay, so these two type of speciation, you can see divergent, which follows speciation, it is called orthologs. So these are called orthologs, frog alpha globin and snake alpha globin, where speciation happens. Okay. So when there's gene duplication happens, we call it paralogs. Divergence follows duplication of a gene. Same gene is duplicated. Okay, and we give two names with a very little difference. So we call it with different names, alpha globin and beta globin. Okay, gene duplication event happened here. So we call it a paralog. Speciation happened here, and we call it two orthologs. Okay, so so that's one thing you need to know, because when you are talking, these terms are important, and when you are reading articles also, these are important for you. And sequence conservation here, you can see. So there are several sequences here: mouse, chick, chimpanzee, human, dog, giraffe, zebra, and the dog. Okay, so those are the species considered, and in this species there are sequences. Okay, so there are sequences 1, 2, 3, 4, 5, 6, 7, 8 given. So what we do here is we are trying to find out similar regions. Okay, and those similar regions in all the five, 1, 2, 3, 4, 5, 6, 7, 8. All the 8 species, we called it a conserved region. Okay, so let me draw this for you. So I think in the classroom normally I use the board, and so what is a conserved region? So assume these are sequences. Okay, And in these sequences, assume these are nucleotide sequences, and you can find several nucleotides are similar. Okay, TG. Okay, so ATG appears in each and every sequence here, 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 and here. So so that region ATG, we call it is conserved in between these 1, 2, 3, 4, 5, 6 sequences. In the 6 sequences, ATG is fully conserved. Okay, and if it is conserved in a set of sequences taken from the whole set, we call it partially conservation. So here, let's see, T, E, G. So it is a partial conserved region. Okay, so this is a partial conservation and this is a full conservation. So, so those are the conserved regions, and what you can do actually is, so when you are aligning these sequences, you can find the conserved regions. Okay. So here also what we do is that, so you can see from this red color plot, it would show you if that is a full conservation in between all the species, it would go to top. Okay, so you can see fully conserved regions from this plot very nicely. Okay, so this sequence is really, really lengthy. So you can see that's why actually it's been chunked and shown. And there are software which you can do this. Okay, integration. And we'll be talking about those software uses algorithms. So in this course, what we are doing is actually we are not trying to apply these algorithms or methods to the biological data to understand biology. Rather than that, our main objective is to understand how these algorithms are formed. Okay, looking into the biological requirement. Okay, so that's our main objective of this course or lesson is. So alignment is key to find important regions. So we talked about that. Why these conserved regions are important? Because we can you say, few people unmute your microphone and answer. Why do you think this finding important regions is useful? What can you do with identifying those conserved regions?
You can identify relationships between different
Yes, we
organisms.
can identify the relationships. That's one. Another, we can determine the function of particular protein. If you can find a similar region of the protein with another protein, which in that protein, if that particular region is responsible for the function of that protein, and if that is been identified earlier, we can say that new protein is also have the same function. Okay, we can predict the function. And we can uncover the evolutionary relationships also. And sequence, so those are the importance. I think now you have some idea about what I am talking about sequence alignment and why it is important, okay? And what are you going to do in this lesson? So those three things should be clear to you. Otherwise, we can't proceed. Can you give me a second? Hello, can you all hear me? Hello?
Yes, madam. Yes, madam.
Can you please get 5 minutes break? I have something to attend, okay? And I'll be here by 5 minutes, okay?
Okay.
Hello? Are you there?
Yes, madam.
Everyone?
Yes, ma'am.
Okay, all the people are here tonight. Okay, so the three things we discussed, what is an alignment? So we'll discuss further about that. But however, you should have uh, idea about what is an alignment now and why is alignment is important and what can you do with these alignments. So those three things should be clear to you now. Okay, so let's have further understanding about the sequence alignment. So we compare DNA or protein sequences mainly for the comparison. What we use it is actually nucleotide sequences in DNA or protein sequences, so amino acid sequences in proteins. So in comparing, we try to find the similarities and the homology. So those two things we look at, okay? So this actually we discussed prediction of function and construction of phylogeny. So these things actually are the separate importance or usefulness or functions of sequence alignment. So why do we do sequence alignment? Okay, so these are the main reasons we do sequence alignment. We try to construct silochaining using sequence alignment. We predict the function of a protein using sequence alignment. And shotgun assembly. So what is shotgun assembly? So shotgun assembly is widely used and widely discussed topic. If you look in the internet, you will find this in many papers and in many websites. So let me erase this. Can you see the whiteboard?
Yes, ma'am.
So what is shotgun assembly? So assume that you are in one end of the world, you are in America. Okay? And in America, you found some, and you shouldn't be in two countries. However, let's assume that you are in America and you found a sequence. Okay? And you are some other person, me, who is in New Zealand. Okay? He also found another sequence, which is of the same species. Okay, so both of these people in these two labs are collaboratively, not collaboratively actually, they don't know one each other. However, they are actually submitting these sequences into the database of a certain particular species. Okay, they are trying to develop the genome of the particular species. So, this one doesn't know that this one has uploaded a sequence, and eventually that the database people, when they compare, how can those people in the central location where these two sequences are submitted find out the real genome, which comes first and which comes second? So is this one coming first in the genome or this one coming second in the genome? So, or else are there anything in between? So how can they find? Can you suggest a way? So like this, there are several labs, people work on the same species to find the genome, okay? And they upload their sequences in central place. And in the people in the central place need to arrange this or assemble this one after the other. So how can it do that? How can they do that? Can you think and tell me something? Hmm?
Take two genes and find the common region.
Yes, that's called shotgun assembly. That's what they do actually. They compare each of these chunks and try to find out whether there are any overlaps. Okay, so if there are any overlaps, they know those are the same parts of genome which have been found by two people. Okay, so they are not repeating that, and they use that section in only one place and they join them together. So that's what called it's called overlap alignments are found, and they are trying to develop the genome from that. And that's one of the purposes, and finding motifs is another purpose. So what are sequence motifs? Sequence motifs are short recurring patterns in DNA that are presumed to have a biological function. Okay, so if you get an particular sequence of a protein, okay, so if you get a particular sequence of a protein, so there may be a sequence motif or a particular short recurring pattern in the DNA, okay, which assume to have a biological function. So this is actually defines the function of that particular protein. this section only. Okay. So often they indicate sequence-specific binding sites, maybe sequence-specific binding sites for proteins such as nucleases and the transcription factors. So that may be actually a region um, uh, specific for a particular function, like activation of that particular gene. So that region is responsible for activating the gene here. Okay. So for the activation, they need certain other protein to come and bind to that. If this protein does not come and bind, this gene would not be activated. Okay. So these regions which have a particular biological function. So what's the function of this particular region? Activation of the gene. So that region is responsible for that. So genes are not always activated because they are not always expressing proteins. Proteins are expressed only when it's required. Okay. So for that, there's a certain region responsible, which is actually this sequence motif, one of the sequence motifs. This is only one example. So those are called short recurring patterns in DNA that have a biological function. Biological function does not intentionally, it does not mean always that protein function. Okay, so this mentions that different functions, like as an example, one function is this. Okay, gene expression. All right. So that's about in sequence alignment, why it is important and where it is used mainly. So finding these motifs also is done using this sequence alignment. Okay, so that's how it is related with alignment. And so we need to look at the sequence variation, because now we are trying to look at the similar regions. In similar regions also, there can be differences. So how does that differences appear? Okay. Differences you can see. There are three main kinds of changes that can occur at any given position in a sequence. One is a mutation, which would replace one character with another. So, assume this is a sequence, and it has the nucleotides as A, A, T, T. Okay, and there are many. I just wrote few. And in a mutation, it would replace one of these characters with the other. Okay, A would disappear and C would come into that place. How can that happen? There are many factors. Okay, that may be some factors from the environment, like radiation. Okay, that's why we are not asked to expose to X-rays more than 2 times a year also. Okay, so there are some regulations because of this. Okay, and you are asked to wear some sunscreen when you go to the sun at noon. Okay, so that's because of those radiation which can be harmful to your skin, which can make skin cancer. Okay, so mutation is something like that, that would replace one character with another. Insertion is that would add one more position. So in insertion, let's assume that we have A, T, T again. In insertion, some character would come here. A, G, T, T. Okay. That would add one position. And in deletion, that would, if you have A, T, T, okay, that would actually delete T. Now we have AAT only. Okay. So these changes you need to know when you are doing alignments, because when you are aligning things, you need to have in your mind that these events would have occurred in these different proteins you get. Okay. So in COVID-19 also, these changes may have occurred because of the medicine we use for the virus, because of the geographical region they are found, because of the environmental factors. These three changes can occur. One of the district changes can occur, and then new strain would develop. Okay? That new strain would be able to survive more than the previous strains. Okay, so that's what the stories are related with this. So the sources of variation. So this variation, I told you, this can occur because of an chemical reaction. Because in our body, all the cells are actually built up of chemicals, and these chemicals would react to each other, and that would change the particular bases into different base. Okay. And replication error. So what is replication? So as I told you, many cells, after they grow, they divide into two cells. Okay. So this is the mother cell, and these are the daughter cells. Mother disappears and two daughters are formed instead. And they grow, and some point they are also divided into two cells. Okay, now this started with one cell, now we have ended up with 4 cells. So that's how the cell division occurs. Okay, so during this cell division process, so this is in nucleus, the copy of the genome DNA appears, and at this stage you can see 2 copies of this. Okay, 2 copies of this appears here. So how can that happen? So this mainly the process of creating 2 copies is called replication. Okay, so this is replicated. This is being copied. So in this cell, actually, in the mother cell, that happens. So this is replicated and then divided similarly among these 2 daughters. So that is actually what happens. So in this replication process, there would be some errors occur. So some places, when it's replicating the same thing, some errors would occur. So the changes would appear because of that. So nucleotide substitutions are actually because of those, mainly those 2 reasons. Maybe a chemical reaction, maybe a replication error. And insertions or deletions, these 2 things. Okay. So they occur because of unequal crossing nova, which is again a process happening in this cell division, crossing nova. So, if you are very interested, you can read this. However, I will just give an introduction. What is this? Because maybe curious to you. So in this nucleus, these chromosomes carry out the proteins. Okay, so the proteins are actually packed in this chromosome. Okay, so they are in spiral shape. They are packed inside this in this as chromosomes. Okay, so crossing over means during this cell division process, when these things are duplicated or replicated, so then some parts of these chromosomes cross over. One of this part, so this part may go to the similar other chromosomes, that part and that part would be interchanged. So this would go to here and this would go to here. Okay, so they interchange. So during this crossover, some parts may be missing. Who knows? These unnatural processes which happen. So unequal crossing over. So this is a longer one, this is a shorter one. So the shorter one go and fit into this, and longer one go come and fit to this. Okay, so insertion appeared here and a deletion appeared here. Okay, so that's called unequal crossing over. In replication, some regions may not be replicated because it's slipped. Okay, in replication, this one. Some regions may not be replicated as we wish. So that's called replication slippage. Some regions may be missed and they are not replicated to their daughters. So that is called a deletion in this particular region. So these variations can occur this because of these sources. Okay, unequal crossing over, replication, chemical reaction, or replication data. Okay, so these chemical reactions mainly happen because of the toxics. So you know sometimes these the food what we eat is sometimes toxic. Okay, so we ban some kind of foods, don't add Ajinomoto. So those are chemicals which can make changes in this body and create cancer. Because you need to know them, otherwise, I mean, as a responsible person, you need to have known these things. So you need to look at what is wrong with Ajinomoto. Why are you not asked to buy the products which sell Ajinomoto? Ajinomoto is not, allowed to sell directly, but you know it's coming in other forms like in that stock. Okay, you all go and buy a soup from the shop, and they say we have not added any Ajinomoto to this. However, at the end, you drink a cup of Ajinomoto because they have added that Ajinomoto to that stock. Because they add stock. They just add some veggies and the stock into water and give it to you. You have to be careful. So those are actually out of the topic. However, I try to give you some idea about them as well because you need to think like that. So those are actually up to now we have talked about what kind of variations you can see in the sequences and the sources of those variations. And now we are going to look at a sequence alignment, real sequence alignment, okay? So what is that? And what things you need to know about the alignments before moving into sequence alignment algorithms, okay? So if you understand this properly, you can actually introduce your own new algorithm for sequence alignment because still there are actually the accuracy is not 100%. Although it's 100%, there are problems. Okay, although it 100% match, we don't know whether it's overaligned. Okay, so there are problems like that. Your seniors have solved problems like that. Okay, so sequence alignment is something like this. There are two set sentences here. So we are trying to identify the similarities. Okay, so so you can see there are gaps here from this minus marks. They show gaps. Okay. Why do you add gaps? To make these two, alignments, two sequences align. When you align these two sequences, why do we need to add these gaps? That's mainly because you know there are indels, insertions and deletions, which happen during the evolution. During these variation, so while happening these variations, insertions and deletions would occur. Okay. So we require these gaps to represent them. Okay. Some deletion occurred here, and some insertion occurred here. Okay. So we would put gaps and try to find out the maximum match. Okay. So as an exercise, you can remove these gaps and try to match these two together. Okay. So you can see the example comes here and heart goes here. And you can't match example with the example in this sentence. Okay. That's mainly because you have not added any gaps. However, in the normal natural situation, those insertions and deletions occur. Okay. And that would reflect, these gaps would reflect those insertions and deletions. So what's a match and what's a mismatch? So you can see when you find similar characters in one sequence, and when you can see the same character in the second sequence, we call it a match. Okay? And we call it a mismatch when you can find different characters in the two sequences. Okay, so it's a mismatch and this is a match. Okay. So if you compare two real sequences, so we are trying to search a series of individual characters that are in the same order in the sequence. Okay. So you can see these two sequences are trying to be matched without gaps. Okay. Can you see any gaps here? No. Okay. So this is an example situation, introductory example situation where you can see any gaps. And they have tried to match as much as sequences possible. Okay, so this T matches with this T here, and this G does not match with A. However, this G matches. Okay, so we would compare two or more sequences by searching for a series of individual characters that are in the same order in the sequence. Okay, so that's what we do in sequence alignment. So if you are asked to define sequence alignment, this is a possible definition. Okay. And so when we are doing this alignment, you may see that some regions are there, but they cannot be aligned properly because we have not put any gaps. In the second situation here, you can see we have put gaps also. So to have a proper definition of sequence alignment, one should include gaps as well. Okay. Given two sequences, X and Y, and alignment is assignment of gaps to positions. This is so that to line up each letter in one sequence with either a letter or a gap in other sequence, so that you can get a maximum number of similarities or similar regions. So this for doing alignments, there are different methods available. Okay. So one of the methods is dot plots, which is being used for years. Okay. Since has a historic, nature, we have to study that here. However, that's actually, there are more better methods than this. So this is the simplest methods for evaluating similarity between two sequences. So to construct a dot plot, so this dot plot is something like this. So you can see here, dot plot is a collection of dots, and these dots represent the similarities. Okay. If one character in this sequence in this axis equals to the character equals to a character in this sequence in this axis, we put a dot here. So that's how we develop a dot plot. Okay. And if there's a very strong similarity in between two, what can you see? There should appear in a diagonal. Okay. You should see a diagonal when you develop this. All the dots in the diagonal are being marked. Okay. So what we do is, the first sequence to be compared is assigned to horizontal axis, and the second is assigned to the vertical axis. So we put the two sequences, one into this and one into this. And dots are then placed in the plot space at each position where both of the sequence elements are identical. So let me draw one for you and this type of thing. It's quite difficult to draw, but I'll try. So here you can see A, T, T, G, T, A, G, G, okay. And another one. Can I rotate this? How to rotate. Okay, let's see. A, T, sorry, T, 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 A. Okay, something like that. What is this? Just draw a box like this. Okay, so what's been done is actually once you can start from here or here. Once you see A here, you put a dot. Okay, and you can see again A here, and you do the same thing this side, A here and A here and A here. A here, there's A here and here. Okay, so like that, you can fill out T here and T here. Okay, so you can see now this diagonal is being formed because A is equal to A and T is equal to T. Okay, which should give you the real similar region at the end. Okay, can you see the whiteboard?
Yes, ma'am.
Okay, so after sometimes I feel like I'm just talking alone and no one is here. Okay, so these plots become overly complex when you compare real two sequences. So which is very have a certain length. Okay, so these plots become complex and crowded. So a plot like this. So that is actually when there are very large number of similarities in between these two sequences, you can see that the plot becomes crowded and complex. Okay, so for that, one suggestion is a sliding window. What's a sliding window? So in algorithms, actually, we talk about sliding windows. Have you ever heard of sliding windows? Hmm? No? So sliding in this window is something like this. So so now we have sequences. What we did was we went one by one, isn't it? We found A and we tried to find A from this side. Okay. So without looking at one by one, we can have a window with having several these letters. Okay. And now we can compare not a single one with a single letter, but a chunk of 4 letters at once. Okay. So here that's what they have done. So without by drawing one by one, you will get a very overly crowded plot like this. However, in here, if you get window size of 10, and what you are doing is we get a threshold. So here, this corresponds to this. Okay. So this window size is 10 and threshold is 3. And window size is 10 and threshold is 5. So here, so previously we tried to draw one by one. Now we are trying to get a box of 10. Okay, the window size is 10 means there are 10 characters in there. And the threshold is 3. So you are comparing this box is with this box. And you need to find at least 3 similarities. And if you find these 3 only, you put a dot. Okay, so now you can see. So here that complexity reduced. And the complexity further reduced by having the threshold 5. Threshold increases and the window size is same. And then only you can see a better comparison in between. I mean, not crowded comparison in between two sequences. So, so there are algorithms to develop dot plots. I think I have given an assignment for that. I don't know. Yes. No, that's not here. Oh yeah, this algorithm is here, so I think you can see it. So given two nucleotide sequences, the goal is to produce the dot plots, dot plot of machine substrings, allowing the user to select values for the window length and searching criteria. Okay. The dot plot produced shows the second sequence on y-axis and first sequence on x-axis. Okay, so this is an example given. So you can produce a small program so that you can do this. Okay. So if anything is not clear about the dot plots, you can read. You should read more about dot plots, and you can look at the techniques they use to reduce the complexities and apply them in your assignment. Okay. And I should actually provide links for this assignment, little tiny assignments in the LMS. I could not. So without sending it to my personal mail, you can then upload this here. So I'll try to do that. So where were we? About dot plots. Okay, so read more about dot plots and read what are the techniques they use to reduce the overly complexness, and look at what are actually important with dot plots and why they are not being used widely. Okay, so let's look at a simple alignment. So to develop an algorithm, you should have a very good understanding about an alignment. Okay, what aspects we need to consider in developing a tool to align two sequences. So let us try to align two short nucleotide sequences like this without considering any gaps. How many possible alignments can you do? Okay. I mean, there's no restriction that you should start from the first letter, match to the first letter of the other sequence. Okay, that's only one way. You can just push one position and then push two positions and try to find a matching region. Okay, so you have now 3 possible alignments. So in the class, actually, I asked you to do this by your own. Then actually it would give me some idea, but now it's not very possible to do here. But I can ask you to share your white screens and do it. But I don't know whether it's successful. So which one do you think is better? Now can you have a look at these 3 and let me know if I mark it as PQR, which one is better? How can you say which one is better? Hmm? Okay, I will give you 1 or 2 minutes. This one is P, this set is P, this set is Q, and this set is R. These are 3 alignments, okay? Alignment P, alignment Q, and alignment R. Which is better?
The P1,
Why?
because it has the most aligned characters.
Yes, highest
Is
number
4.
of
It is 4.
similarities.
Yes.
Yeah, there are 4. How many are here? There are none here. There are only 3 here. Yes, you can say it's P. However, to find a very good alignment, do you think um, this kind of aligning is enough? I don't think so, because sometimes there may be regions which we miss because we have to put these 2 things in a row, okay, without having any gaps, in other words. Okay, with having gaps, we could go for a better score or a value, a higher number of matches. Okay. Nusrat lecture එකේ ඉන්නේ. ඔව්. මාසේ කතා කරන්නේ. නෑ මම ගෙදර ඉන්නේ. ඔව්. So you can see by inputting gaps, we could be able to find more matches. Okay. So as the second step, they try to add gaps. Apart from that reason, to find highest number of gaps, you can actually think this in another way. That if you look at any 2 sequences, the variations can appear as indels, insertions or deletions. Indels. Okay. So these insertions and deletions can be represented using these gaps. Okay. So there are 6 matches here, 3 mismatches on 1 gap. So here, after including gaps, we can see 7 matches, 1 mismatch, and 3 gaps. And here there are 7 matches, 0 mismatches, and 5 gaps. Okay. So after we add gaps, also that it's quite difficult to define what is the better one, isn't it? Although there are a higher number of matches here, there are lower number of gaps here. And there's 1 mismatch here, there are 0 mismatches. So how can one decide what's the best alignment between these 2 sequences? So that's another problem which appears. So what's the solution? We should have a scoring technique. Okay, we should consider match score, mismatch score. It means number of matches. We should consider the number of matches and number of mismatches and number of gaps. Okay. So we need to have a scoring mechanism to evaluate the alignment. Okay, we need to look at the matches and mismatches, and we should define how many marks are we going to give for matches. If we find a match in 2 sequences, what mark or score are we going to give that match? And if there's a mismatch, what mark or score are we going to give? We have to decide on that if you want to define a score mechanism. And we can have the total score as this if we consider these matches and mismatches. Okay. You should count the number of matches and you should count the number of mismatches, and you should multiply the number of matches with the match score, and you should multiply the number of mismatches with the mismatch score. Okay, and then you have to add them together. Okay, so by adding them together, you can have a total score. Okay, so here for the same example, they have achieved the match score as 1 and mismatch score as 0. And as we previously looked, we can now have match score, score as 4 here, 1 here, and 3 here. Okay. So, however, we know now we require gaps, isn't it? Good aligning means we have to consider everything, insertions, deletions, and we should try to maximize the score of that alignment. So for that, we need gaps. So how can you consider gaps also into our scoring scheme? Okay, so here they have used gaps and they have aligned it here. So considering the match score as 1 and mismatch score as 0 and a gap as -1, what are the scores of these 3 sets? Can you find out that now and let me know? So this is P, this is Q, and this is R. What are the scores of P, Q, and R considering a gap score as -1, match score as 1, and mismatch score as 0? Can you let me know now? What's the score for the first one?
One.
Yes, it's 1. For the second one?
Still doing.
What's that?
Still calculating.
Okay, okay.
Three.
3. And the next one?
Three.
Yes. I think you all got it. And if there are any problems, please ask me. So a scoring function can be defined now. So what you have done is written as a standard function using some standard terms. Okay, if we find a match and the score is plus n, and if you find a mismatch, the score is minus s, and if you find a gap, the gap score is minus d. Okay, so what you have to do in scoring function is the score f equals to number of matches multiplied by m and subtract by number of mismatches multiplied by s, and again this is actually the negative sign comes from this scoring value negative. Okay, number of gaps multiplied by d. D is the gap score. Okay, the negative sign of d has appeared here. So so if you look at this, so this gap penalty, so you can see here, okay? So the gaps can be at the end. So here the gaps are not marked. So here, gaps can be at the end as well. Gaps can be at the start of a sequence. They can be at the start, they can be at the end, or they can be at the middle. So do you think having the same score for the gaps at the end, at the start, or at the middle is good? They have found that it's not good mainly because nature favors small number of long gaps compared to large number of short gaps. Okay. Apart from that, there may be very lengthy gaps. So here, there's only the length is only 1. However, we can have very lengthy gaps also like this. Okay, A. And again you have gaps, very lengthy gap. G. Okay, you have another one, something like this. A, G, G, G. Okay. So I give it the same score for this gap and for this gap. So with this alignment of the gap with the element. However, this is a gap. So when the gap is there, you have to apply the gap score. So are you going to apply the same value for the gap score here, 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 and here? Okay, is it a good way to do that? No, because actually good alignments have a small number of long gaps. Okay, that's actually a decision taken looking at real natural sequences. Okay, so when you're provided 2 sequences to align, what you have to do is you have to align them so that you can biologically have a meaning. Okay? So we should consider biological observations. So biologically, they have observed that nature it would favor small number of long gaps. There are no very lengthy gaps. Okay, and there are large number of shorter gaps. The nature favors small number of long gaps compared to large number of short. So sorry, I think I misled you. So nature would always favor a small number of very long gaps rather than having short gaps here and there. Okay. So, so that those observations also should be incorporated with your gap score. Okay, so this is not all the observation. There are more. This is one example. So how do we adjust our scoring scheme? Take in this fact also. So we may have different gap opening penalties. So this is a gap opening place, isn't it? And closing penalties. Okay. So this is the gap open. So we may give a score, and after the gap score, we may give for the second gap position another score. Okay. So the gap score may be very high negative value, okay, because there are no much small gaps on them. Okay. Rarely you can find a few. However, many of the time the nature favors long gaps. So we can avoid giving a high score for a sequence like which have many short gaps. We should give a lower score for that kind of alignment because nature does not allow that to happen always. Okay. In nature, we can't see such kind of alignments always. So to find that kind of alignment, one can have a technique in scoring. Okay, like that. So we can have origination penalty. Start in a new series of gaps. In one of the sequence, we allow origination penalty. Gap starting penalty. Okay, opening penalty. And length penalty depends on the number of sequential missing characters. So when the gap becomes lengthy and lengthy, we assign the score in ascending order. We increase the penalty which we put for each and every gap which appears one after the other, so that we can have a less score for that alignment. Okay. Having a less score means it's not a good alignment. It does not tally with the nature, naturally found alignment. Okay. So you can change the scoring systems according to the got the observations. Okay, so apart from that, there is scoring schemes actually. So in our scoring scheme, which we discussed,
Excuse me, madam.
which we here, so as an example, if I draw two alignments, so these are the two alignments, and you would give the max score only when you would find the same element in the two rows. You would find A with A, okay, T with T. If you would find only this, you will give the max score. Okay. You would give the max score only when you see the same element in the two rows. Okay. However, this biologist has found out that apart from the similar characters, the second character can also be another one which have similar chemical characteristics with the first one. Okay, so this mainly occurs when you are comparing proteins. Okay. If a protein in the first sequence have a very high similarity of the chemical characteristics of another protein, you can also give the max score for that. Okay, that's one specific place we should consider. Okay. So because of this reason, they have introduced scoring matrices. Okay. Instead of having a single match mismatch score like this, for every pair of nucleotides or amino acids, consider chemical, physical, evolutionary relationships. Okay. And examples are alanine and valine or alanine versus lysine. Alanine and valine are both small and hydrophobic, but lysine is large and charged. With substitutions occur more in nature. Some substitutions may occur frequently in nature. So those are also considered this in this scoring matrix. So considering these chemical, physical, evolutionary relationships, they have assigned scores to each pair of symbol. Higher the score, higher the similarity. There are the two main substitution matrices used, Ham matrix and Blossom matrix for proteins. Both try to capture relative substitutability of amino acid pairs in evolution. Okay. So here you can see Blossom 62 matrix. So there you can see in this side you can see proteins, and this side also again the same set of proteins. And if you find this pair, what would be this negative 2? If you find this here, I with I, okay, so that's actually obvious. So here W with W, it has the high weight because it's rare. Okay, that's a very good match. And here E with E, 5, sorry. E with G, negative 2. So likewise, you can see some odd places as well. So this is actually developed considering those not evolution relationships. Okay. So this is Blossom. And you can look at PAM also in the web. Okay. And read about those matrices and read how you can identify, use these in your algorithms. So now we are going to look at the interesting part for computer scientists, which is dynamic programming. How can you find the alignments? Okay. So when you are using dynamic programming to find a better algorithm to solve this alignment problem, so you have to look at different alignment types. First, we are going to look at global sequence alignment. Okay. So global sequence alignment means we are trying to align the whole sequence length with the second sequence. Okay? The entire length of two sequences are considered. Okay. So, so when this is suitable when the two sequences are of similar length with a significant degree of similarity. Okay. So as an example, we are trying to match this corner with this corner. Okay. So we have put some gaps. That's okay. However, we are considering the whole length. This with this. So what can we do to find the best alignment? So the first one is brute force search. So you can you can write many combinations as possible. You have to put gaps, okay? Similarity has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Similarity has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 positions. So you have to actually compare the 7 positions with 7 positions of the second one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this 10, sorry, not 10, 10 positions, or the length is 10. So you have to consider 10 as here by applying 4 gaps. So that's what we say the entire length is aligned. Okay. You should have same number of positions in the second sequence as well by putting gaps. It's okay. Okay. So when it is suitable when the sequences are similar length, because then you may not be want to put many gaps here. So how do you find out the best alignment? Okay, we can draw different, we cannot draw actually, we can align this in different ways which we can do, okay, manually or whatever it is. You can, this is a one way. There may be many other ways you can put these two together, okay, with having 4 gaps in the second sequence. You can put the 4 gaps in front, and you can put the 4 gaps in back, and you can put some gaps in front and the other gaps inside the sequence too. Likewise, you can create many alignments, and then give us apply a scoring system like we discussed previously. For match score, you can assume some score value, and for mismatch score, you can assume some different score value. So likewise, you can actually take a score for the alignment and then find the highest score, and that would be actually the best aligned two sequences with gaps at the particular positions. So that is called brute force approach. Generate the list of all possible alignments between two sequences and score them and select the alignment with the best score. So that's called brute force approach. Okay. Brute force approach. That's a straightforward approach we always think of. Okay. However, when the length increases, what would happen? Using this formula, you can have this number of possible alignment pairs, which is actually, even with the computer, impossible. Okay. So, however, people have observed some characteristics of these alignments and developed dynamic programming methods for this problem. Okay. So the characteristics they observe, the first characteristic is additiveness of the alignments. Okay, we can get chunks of these alignments, and then we can add the scores together. Okay, so that's the additivity of alignments. Okay, so that's one thing they consider. And secondly, no, that's the actually thing they consider, and depending on this additivity, they develop these dynamic programming algorithms, because we can take chunks and calculate the score and then add together. Okay. So if you consider these two sequences, CA, CGA, and CGA, how many possible ways of aligning this? So you can align in first position, you can have two elements, not C, not actually considerably. And I mean, it should be not be C. Here you have A and G. Okay. Sorry, here you have C's. Yeah, they have considered that C. You can align C with C. Okay. You can align C to C, can't you? You can get it here. C to C. So the score is +1 and the remaining sequences are these. And then you can put a gap to the first sequence and drag the second sequence under the gap. Okay, now you have, so like this, C, A, C, G, A. And the first time what we did C, G, A like this. The first position is written here. Okay. And then we can move this one a bit this side and develop something like this. A gap, C, A, C, G, A. Okay. And then we are not moving this anywhere. C, G, A. So here you can see in the first position now we have gap and a C. Okay. So for the first one we have two elements. The second one we have gap and an element. And the third possibility for the first column is you can have gap in the second sequence. C, A, C, G, A, and there's a gap here. C, G, A. Now you can see at the first question, there are three main possibilities and three scores, and these are the remaining sequences. So in dynamic programming, this kind of possibilities are considered and the scores are calculated, and looking at the additivity of an alignment, they are added. Okay, so that's what is done in dynamic programming of sequence alignment. So these are the three main possibilities. We can place a gap in the first sequence, a gap in the second sequence, or we can place a gap in neither sequences. We have what? Elements. Okay? Alignment score for 1 and 2 is gap penalty. If you see this, 2 and 3 is the gap penalty because we can see a gap. And alignment score for the other depend on the match or mismatch. So if this is a match, it would get the match score. If it's a mismatch, it would get the mismatch score. So there are three things. Okay. Either we can have a match score or a mismatch score or a gap. Okay. So the first algorithm so this is one of the cornerstones of bioinformatics, the Needleman-English algorithm. Okay. So looking at these characteristics of alignments, they have developed this algorithm. Okay. So let's have a look at this, how this is being done. Now we know all the characteristics. So these are the main steps of the Needleman and Wunsch. So, so let's have a look at this and try to develop this. So Needleman and Wunsch algorithm. So what you have to do is actually you have to develop a matrix first and then put the two sequences at two sides. Okay. And you should have a scoring system. And in this scoring system, you should have gap penalty value, match bonus, and mismatch score. Okay, in this particular example, we have taken -1, +1, and 0 as the score values. So here you are assuming two gaps initially. So that's a must when you want to align two sequences. And you put the two sequences on two sides. So the initialization is the first step. Initialization of the scoring matrix. So that's done like this. You consider this as a gap. Okay. Gap and gap, the score is 0. And gap and A, so there's a gap in the first sequence and a value in the second sequence. We put the gap penalty here. And now we see another gap here and another element here. However, so since these alignments are additive, we have to add the previous value for the gap penalty you see here. You get actually for this combination C and C in the first sequence and a gap in the second sequence -1 because gap penalty is -1. -1 is added to -1. This has the value -1. This is with this one actually. However, this -1 is added to -1 because these are additive. So you get -2. Likewise, you get -3, -4, -5 by adding the score to the previous one. Okay. And similarly here, there's a gap here and an element here. You put the negative value. Okay. And then there's a gap here. Again, an element here. The score is -1. However, you have to add the previous gap penalty. Sorry, previous score, mainly because these are additive. So likewise, you can initialize the matrix. So after that, how can you develop the matrix? So in this algorithm, in each step, actually, these steps are actually summarized here, okay, in general. So in this, we have done up to this. We have done up to here. And now we have to calculate the scores. Horizontal move is a gap at the left sequence, and vertical move is a gap at the right sequence, and the diagonal move represents an alignment of the nucleotide from each sequence. Okay. So can you remember that we discussed about three possibilities, isn't it? Gap at the first sequence, gap at the second sequence, and values from two sequences. Actually, these three things are considered in this step, isn't it? So how can you represent that in here? Okay, horizontal move. If you are going from here to here, so it means it's a gap score. What's the gap score here? -1. So I'll write that here. I erase those things. Okay, so if this horizontal move, it means a gap score. Okay, I'll just write it -1 here. And if it's a vertical move, it's also a gap score. So I'll write it here. So, okay, so this is the vertical move, and this is the horizontal move. Okay, the other one. So if you come this way, it means diagonal move. So these are three possible ways to assume that you can come this way, or this way. So this is either match, mismatch, or match. Okay, it can be either 1 or 0. Okay, so what we are doing here is, you see here 0 is here, and -1, -2, -3, 4, 5, that those are initialization values. And here you come here and see what is here. So there's A matched with A. Okay, 1 is the value here. So that one value you can come from 3 sides. Okay, so you are going to get the maximum out of 3. So these are the 3 ways you can approach this cell by having these 3 possibilities at the previous position. Okay, so it's something like this. So in the previous, we had 2 sequences like this now. Okay, now so there are 3 possibilities. We can align like this, or we can have a gap and C, C, G, A, T, and C, G, A. And there can be a gap here, and C, G, A, T, and C, G, A. Okay, so these are the 3 possibilities. So the first position, either you may calculate this by happening this one for the second position, for the first position. Now you are here, you need to look at the second position, second alignment, okay? Alignment for the second window or the position. So this is the first position, okay? So when you are doing the alignment for the second position, these 3 possibilities may have happened in the first position, isn't it? Okay, Needleman and Wunsch algorithm. So when you are scoring the second position, you should think about the possibilities that could have happened in the first position. So these are the 3 main possibilities, and these are those 3. This, and this. Okay, so you have to consider all those 3 possibilities when you are coming to this position. That's why I told you to calculate the score for this position. It means the second position. You should consider coming from this way, and this way. This way means there's a gap in one of the sequences. This way means there's a one of gaps in the other sequence. And this way means there's no any gaps. There are elements in both sequences. This situation. So when you are considering a score for the second pair, you should consider all the possibilities for the first pair. And you have to consider the maximum score from these 3. So how can you do that? Okay. So you come from this side. So this side means there's a gap in one sequence and A in the other sequence. So the score is -1. And when you come from this side, horizontally, it means there's a gap in one sequence and A in other sequence. Again, the score is -1. And when you come to this position, if all the 2 sequences have elements, it means It means what? There's a match score of 1. Okay. 1 is here. 1. Okay, I'll write it here. So now for the second position also we have look at the possibilities. 3 possibilities. Okay. There's a gap in the first sequence, and there's a gap in the second sequence, and there are elements in the G and G. These 3 possibilities scoring values are marked here: -1, -1, and 1. This is 1, -1, -1. However, when you are considering these 3 possibilities, you have to consider for each one the previous possibility as well. Okay. So for that, you need to add the previous value to this one. -1 added to -1. What is the value? -2. Okay. And here, 0 added to 1, it equals to 1. -1 added to -1, this way comes from this side, it is -2. Now from these 3, -2, -2, and 1, so those were the possibilities. You have to find the maximum score. So that's the winning score. We are not considering this one or this one. So it means we have not come from these 2 ways, and we have came from this way. So you should draw the arrow like this. You should not draw the other arrows, because you have get the score, maximum score value from in this way. Okay, having C and C in the previous position. So likewise, you have to fill out all the positions in this matrix. So let's go to the second next position. It means this position. Okay. So when you are considering the score for this, okay, you have to consider all the previous alignment situations. These 3 here and these 3 here. So for that, so now we have get the maximum value to this one. Okay, so you do not go to here. Rather than you have now discarded the other 2 possibilities and come up with the highest value. Okay. Now when you are looking at this position, you have to consider 2 sides, 3 sides, 1, 2, and 3. So here you have to consider there's a gap in the first sequence. So we put -1 here, and gap in the second sequence, we put -1 here. You are coming from here, C and A, there's a mismatch. Mismatch score is, I can't remember, 0. Okay, 0 here. Okay, now you have here -1, -1, and 0. You have to add those values to previous scores. -1 into -2, -3 coming from this side. Coming from this side, -1 into +1, 0 coming from this side. Coming from this side, C to A is 0, and it is added to -1, again -1. So what is bigger? -3, -1, or 0? 0 is the bigger one. So you put that value as the score. Okay, so likewise you have to fill out the matrix. So I'll give it as a homework and stop the lecture for now. So you have to practice doing this. Okay. So you should have some understanding about what's happening also, and try to fill out this, and then we will at the next step, what we are doing is we use this matrix and try to find the best matching sequence chunks. Okay, we are trying to find the best match of 2 sequences, or best alignment of the 2 sequences. So for that, actually you need to draw these arrows. Okay, whatever the arrow is you use to come to a score in this box. You use this arrow to come to us. and you use this arrow only to come to here to get the value here. The other arrows don't let them there. Only these arrows where it will show you the direction where you come from. Okay. So that should only appear in your matrix. Okay, I'll stop for now. Have a look at some material about this pairwise alignments using Needleman and Bush algorithm, and try to fill out this table. While filling out this table, you can see the answer this side and check whether you are doing it correctly. And apart from that, you need to do the last exercise where you would have to develop a dot plot by your own. Okay, so I'll stop for now. Are there any questions? Are you there?
Yes, ma'am.
Okay. Now I'll stop the lecture and upload material to the LMS. And I'll upload 2 assignments, upload links as well. 3 assignment upload links for 3 weeks. So then you can upload 3 or 2. I think there were 2 exercises plus this one. Yes. So this lecture is the second one or third one? Third one, say.
Yes, third.
Okay. So I'll put the upload links for the 3 lectures. Okay, thank you very much. Have a nice
Okay, thank you, ma'am.
Thank you.